Good morning. What I want to do today is do a quick overview of Mendel's genetics, his pea plant cross, and how that's going to relate to in the future as we keep going. Mendel had been a monk in a monastery, lots of free time, and did huge numbers of experiments regarding crossing pea plants and the traits and how the traits descend from the parent plants to the son and daughter plants. So, first we got to remember that when we cross two animals, it's a process called fertilization. You have half of the DNA comes from the mother, and exactly half comes from the father. But when we see humans crossed and other animals crossed, we notice that some traits will show up from the father, some show up from the mother. But it's important to remember, even though you may have dad's eyes, you have a copy of mom's eye gene, one or the other, that she got from your grandmother or her grandfather, your grandfather. So, when we cross pea plants, we're going to talk about an organism that is purebred versus a hybrid. These are different from the normal uses, such as in horse raiding or dog breeding, of a purebred versus a hybrid. When we talk about a purebred, in Mendel's conception, that's for one trait. If he has a pure, tall plant, it will only make tall offspring, regardless of the cross or the odds or anything like that, because it has two tall alleles. It doesn't carry a short, so it can't produce a short offspring. Now, so a purebred would be tall, tall, or short, short, because everybody has two copies of their DNA. One copy says tall, the other copy says tall. Just like in the short plant, one copy says short and the other copy says short. Notice that I used little t's to start the short. There's a real good reason for that we'll look at in a second. So that's a purebred versus a hybrid. So we're going to talk about the F1 generation. These are the first children of our test cross. So if we cross a tall plant times another tall plant, those are the P generation. Their offspring are the F1 generation. If we cross those F1s, that's the F2 generation. From philia, that's what the F stands for, filial, which means family. So, Mendel crossed a tall plant times a short plant, and then of all the plants that came out, every single daughter plant was tall. Draw out a quick family tree here. So even though there is a short parent, all the plants are tall. This is because even though every one of them had to get a short gene from the other parent, the tall gene over here is dominant. Because remember that this guy has two big T's, this guy has two little t's, and the way we show dominance is by having a big letter. And whenever we say these letters, you always say big T or little t, never just T, because in genetics it's really important that we know if it's a dominant trait or a recessive trait. So we always use lowercase letters for recessive. Recessive means it can be covered up or hidden by a tall or a dominant trait. Okay, so if we look, we get one from this parent, one allele or gene from this parent, which means all these children have one of each. So I can go through and mark each of them, big T and little t, because every offspring in this F1 generation has a mix. That's what we call hybrids in this Mendel study. There are a big T and a little t. Now if we cross two of these, a big T and little t plant, then we start to see the effects of this dominance or recessive come up. We will usually get 1, 2, 3 dominant trait plants and one that shows the recessive. Because if we mix the big T's and the little t's across, one from each parent here, one from each parent here, one offspring gets both little t's, and then the next offspring gets little and big, then we see that we get 1, 2, 3 talls. But notice they don't have all the same genetics, but because they're all uppercase, they show as tall. And then one short, because to be short, you have to have two little t's. So that's where we get the 3 tall to 1 short ratio in this cross here. So that's just a rough beginning of that. Then Mendel experimented with other traits, such as the color of the peas, whether they were wrinkly or smooth, and was able to find out which were dominant and which were recessive. And his tests usually involved a couple thousand different plants, a couple thousand offspring at a time. Now, so dominance and recessive, the two versions of that gene we call alleles. It may be a gene for tallness. The different alleles or versions of it are tall or short. Just like my hair, there's a gene that makes hair. Different alleles of that gene or versions of that gene will make my hair dark or light or curly or straight. Those are all just versions of the hair recipe, but it's still the same basic recipe for hair. So a gene is the overall gene for hair. An allele is a different version of that gene, which makes a slightly different recipe. Still hair, but a different color. Still a pea plant, but a short one instead of a tall one. Now, dominant allele will always show if it's present. You have to have 2 recessives before it'll show a recessive trait. Now, in Mendel's crosses, he referred to these as purebred and pure short, but these would all be hybrids. In horse mixing or something like that, a purebred is an entirely different idea. And we're going to switch to different words very soon. This will be homozygous, homo meaning same, 2 of the same alleles, or heterozygous, 2 different, for hetero meaning different. So these will be homozygous, tall, homozygous short, or heterozygous. We don't need to say tall or short with heterozygous because a heterozygous always shows the trait of the dominant allele. In this case, all heterozygous or hybrid organisms will be tall because they have 1 tall and 1 short, and tall is dominant. Now, Mendel's contributions were extremely important, but also he was extremely lucky because a lot of genetics interactions are not as simple. This is the most simple. It's just called simple dominance. One is dominant, the other trait is recessive. Dominants always cover recessives. Quick recap, we always have an uppercase letter for the dominant, lowercase letter for the recessive, and we always say big T or little t, big Z or little z. Never just Z, because which one it is really important in this study. And then finally, when you're writing them, always put the big letter first, because big T, little t is the same as little t, big T, but it looks different. So when we're counting totals on these, they need to all look the same. So big letter always first. And we'll do a whole lot more on the basics of genetics very, very soon.